I apologize for the last one, got cut off there, but I wanted to finish up taking a look at some of these other models. And when we look at this process of attenuation, the folds within the RNA itself are actually going to be controlling translation of the enzymes that are involved in trip biosynthesis. So as long as there's plenty of this amino acid around, your ribosome is not going to be able to make your proteins. And once your trip gets used up, then this thing is going to switch over to where transcription and translation by your polyribosomes can continue. But again, it's only under these conditions where the cell is low in trip. So this is a really important idea that there's multiple levels of control that have evolved in these cells, and that shows you how important this process is to saving the cell's energy. And when we look at this, we are looking at something that people have called the riboswitch. And here, the switch is built into the RNA, and it can actually affect whether or not things are used, because the message actually will fold up into a three-dimensional shape that will actually bind to a ligand or some sort of molecule out there. So in this case, our RNA is effectively acting like an enzyme almost, with an active site that can bind to something. And so if we look at this case over here on the left side of this figure, you've got this in the on switch. The RNA is folded up in such a way that the RNA polymerase is going to be able to read through, and you can get your transcription to proceed. But over here, when you get this ligand, this little molecule is able to bind to that chunk of RNA right there. Notice the stem loop that we see is broken right there, and that changes the complementary bases that can bind to this little guy over here. And when you get those changes in the shape of your message right here, this can actually result in your message being destabilized. So this RNA is going to be broken down more easily, and that can result in the lack of an enzyme being made. And so if you imagine if this is for some sort of biosynthetic enzyme that produces this ligand, well, here the ligand, instead of going and binding to the enzyme, it's actually binding to the message and regulating translation of the protein that would be involved in its synthesis. And as long as this ligand isn't present, you're going to have the transcription and translation, so that you will actually get your enzyme that can produce the ligand to turn it to the off side there. So your switch is literally built into the RNA because of the three-dimensional folding there. In this example, we see something similar, but here you're actually doing control at the translation level here. So there you've got your message right there. Your ribosome can go ahead and bind. We've got this ligand that binds to your message right there, and in this case, it changes the shape of your message so the ribosome can no longer bind. So imagine that you've got your AUG right there. Over here, it's able to bind, or the ribosome can bind and start doing translation, but over here, the AUG is bound up, so the tRNA has nothing to bind to, so you shut off translation because of the shape of your RNA. Now, all of this stuff, we've been looking at different models that were determined using prokaryotic cells, which you remember, haploid, circular chromosomes, everything is in the same little bag there. Transcription and translation occurs in the cytoplasm. But when we look at eukaryotic cells, now we see a whole different beast altogether, and it gets a lot more complicated. When we look at regulation of gene expression in eukaryotic cells, the RNA polymerase is actually made up of 40 to 50 different proteins. So we're not talking about your core polymerase with a sigma factor. Now we're talking about potentially 20 or 30 different kinds of sigma factors that are all globbing onto this thing to change how it works. And we've got this additional thing, is that eukaryotic DNA can actually fold or bend over on itself, and these distal elements, so things that are close by, but they can actually be pretty far away from the promoter. If the DNA folds over correctly, it brings this sequence of DNA close to the promoter, and that would be your equivalent to like your sigma factors. Multiple different activators could bind to trigger your RNA polymerase to go ahead and do the transcription. And if you change that distal control elements, like in this case, you can change which gene is being transcribed. So a lot more complexity in the situation, because not only do you have enhancers, you can also have repressors of transcription. So you're looking at multiple different combinations of factors that can come into play and really fine-tune your gene expression there. And as if that isn't enough, there's another thing out there that's known as epigenetic regulation. And when you look at these guys, they can have multiple effects, and they think those effects can even be transmitted from one generation to the next. So think of it like instinct. These things are built into you from the minute that you're born. And what these rely on is actually changes to the chromosome molecules themselves. In some cases, you will actually add a methyl group to your DNA, and that can change whether or not the DNA is actually used. And in other cases, they will actually take the histone proteins that your DNA is wrapped around, and they will modify those histone proteins by adding acetyl groups. So 2 carbons attached to those things can actually change your structure of your DNA there. And if your DNA is wound up tightly, that gene is inactive. Whereas over here, because of some sort of epigenetic factor, whether it's a methyl group or adding acetyl groups to your DNA histones, that can result in your structure of the DNA changing, and now your gene is active. So a lot of stuff is going on, and people have even found cases where the location of the chromosome inside the nucleus can affect whether or not transcription occurred. And they found this in a case that I read about where if you have a chromosome that is towards the periphery of your nucleus, your genes tend to be silent or less active there. And they found a case of somebody who had a disease where their chromosome was on the periphery of the nucleus, the genes were not turned on, and they got this phenotype. Normally, people who don't have this disease, that particular chunk of the chromosome was towards the middle of the chromosome where the DNA was more active. So not only do we have structures within the DNA, we have structures within the messages, and we've got this location of your DNA. So all over the place, we can see that this control of gene expression is extremely important for the health of our cells that are out there. And hopefully you have a new appreciation for a lot of what's going on over these billions of years, where Mother Nature has been really rolling the dice, and every once in a while the cells win, and they evolve something that is actually going to allow them to perform things better than what their siblings are able to do, the other members of that population.